The overall goal of the following experiment is to apply this in vitro dormancy model to study molecular and cellular mechanisms and to generate hypotheses for testing in more biologically complex in vivo models. This is achieved by culturing estrogen-positive breast cancer cells in fibronectin-coated tissue culture plates at clonogenic density, where their interactions are primarily with the substratum and not each other. As a second step, a dormant state is activated in some of the estrogen-positive cells, supported and maintained by FGF2 and fibronectin, representative of breast cancer cells in the bone marrow. Next, the system is perturbed by addition of antibodies, inhibitors, peptides, or nucleic acids in order to disrupt various phenotypic and molecular aspects of the dormant cells and determine their role in the maintenance of the dormant state. The results show the number and appearance of growing and dormant colonies that are the results of treatment of incubated cells with FGF2 on fibronectin-coated plates.
This method can help answer which signaling pathways, adhesion molecules, or other specific molecular mechanisms are responsible for development and maintenance of dormancy in estrogen-dependent breast cancer cells located in the bone marrow microenvironment.
To begin, aspirate the culture medium from plates that are approximately 50% confluent with MCF7 cell lines and rinse them with pre-warmed PBS. Then, add 1 milliliter of 0.25 trypsin EDTA solution dissolved in DMEM high glucose medium with